This is a review of the evidence base for trigger points. The evidence base is incredibly important, or increasingly important, in modern medicine, and it is based on studies that are done in the literature. So what these studies do is that they give credibility to trigger points in the mainstream of medicine. Our story begins with a nerve and a muscle. Message comes down the nerve, and it comes down to these little fingers, which are actually called motor end plates. The motor end plate then is blown up across here. So now the message is coming down to the motor end plate, and it strikes these tiny little sacs. These sacs are full of acetylcholine. They degranulate, and the acetylcholine moves across the space. It's called the synaptic cleft, and it attaches to these receptors, and now that message from the nerve has been transmitted to the muscle, and it will tell the muscle to contract. So the more acetylcholine sent across the cleft, the more the muscle will contract. Now, the fascinating thing is that you can measure the amount of acetylcholine moving across by the electrical activity that it induces in the muscle. And this is the first study that we're going to look at. In 2004, David Simons, who is one of the main workers in the field of trigger points and who wrote the red Bible of trigger points with Janet Travell, in 2004, David Simons did this study. Now, what he was doing was he had a very fine little needle which he put into a trigger point. And what he was doing with this fine little needle was he was measuring the electrical activity at the motor end plate. And if you look at this, which is A, he is showing each of these tiny little blips. These tiny little blips is something called a miniature end plate potential. So it's a tiny little package of a little electrical current, which is showing a tiny little package of acetylcholine going across from the nerve to the muscle, and this is a resting human muscle. He did it also with a rat, so you can see they're very similar. He then showed that when you stimulate the muscle, you get a picture like this. So the nerve sends many more messages, and you can see that there's a tremendous difference between A and C. And D is similar. It's a complex pattern. However, if we look at E is the very significant finding. And what you see here are miniature end plate potentials, which are completely different from C. And E is the measurement of a muscle with trigger points. A is a measurement of a normal muscle at rest. And so you can see there's a tremendous difference between the electrical activity of a muscle at rest and of a muscle with trigger points. This is called spontaneous electric activity, shortened to SEA. Originally, the workers in this field, electron myography, considered this to be just some kind of interference. But what David Simons showed is that this occurs at trigger points. If you move just half a centimeter away from the trigger point, you get that picture. And this kind of activity is showing that there's a tremendous amount, there's increased amount of acetylcholine going across, causing excess contraction of the muscle which is being supplied purely, the part of the muscle that's being supplied purely by those little motor end plates in that area of the muscle. A further study by Hubbard and Burkov actually measured the amount of acetylcholine and showed that there's increased release of acetylcholine with trigger points. And this is a very famous micrograph. So this is a photograph of a section of muscle as seen under a microscope. This is normal muscle, and you can see these very fine lines which I describe in greater detail in the ultrastructure of muscle. Here is a small area within the muscle, and if you look at it, it's quite different from the surrounding area. The lines are much closer together, and this represents a so-called tiny little contraction knot within the muscle. Now, this area would have been stimulated by excess acetylcholine production from one or two or three of those tiny little motor end plates that we looked at just before. We are continuing on from the contraction knot, which we saw. This is a picture showing the kind of distribution that the contraction knots occur in. Now, in 1988, Haag developed a fascinating hypothesis, which he called the Cinderella hypothesis. In this, he postulated that the pain of trigger points, and he actually was looking specifically at occupational muscle pain, he said that this is caused by selective overloading of the earliest recruited and the latest de-recruited fibers in muscle. Now, you know that there are two types of muscle fibers. There are type 1, which are the grunty muscles. These are the muscles that don't require a lot of energy, so they don't have a lot of mitochondria, and they need to do low-level work. These are the muscles that are very, very important in posture and in repetitive work. These muscle fibers, the type 1 muscle fibers, occur more toward the end of muscles, and they are closer to where the muscle joins into the tendon. The type 2 muscle fibers are much, much more energy hungry. They are the power muscles. They tend to occur closer to the belly of the muscle. And with trigger points, you have selective recruitment of the type 1 muscle fibers. So these muscle fibers are continually stimulated by the acetylcholine that we talked about before. These little contraction knots tend to be more of the type 1 muscle fibers, and so these become the Cinderella fibers that do all the work, while the ugly sisters, which are the type 2 fibers, sit around and don't do very much at all. Several newer studies have actually confirmed the Cinderella hypothesis and support this idea that these low-level static exertions, which is really holding a posture, sitting in front of a computer screen and doing keyboarding things in, working with a mouse, all these kinds of movements preferentially stimulate the type 1 fibers, which set off contraction knots and set off trigger points. And there were two really interesting recent studies. There were two recent studies, the first by Tristas in 2006, where he looked at a number of people doing just finger tapping on a keyboard in front of a computer, and he measured whether they had trigger points turned on, and it required just 30 minutes for the majority of people to turn trigger points on in posture muscles and muscles that were used in this activity. And then Chen in 2000 took a group of piano players, and this time it took only 20 minutes for them to turn on trigger points. And what they were doing was they were preferentially turning on or stimulating the Cinderella fibers in the muscles that were being studied. In 2006, Otten studied the pressure gradients in a gastrocnemius muscle. This was in a toad, but these are transferable to the human gastrocnemius muscle. And what he showed was that the pressure is much, much higher at the tip, at the ends of the muscle. And he confirmed that during static, low-level muscle contraction, capillary pressures increase dramatically toward the muscle insertions. Now we know that the Cinderella fibers live mainly at the muscle insertions and at the end of the muscle. And these are the areas where trigger points occur. And therefore what you see is, if you have an area of muscle which is full of these little contraction knots, you get excessive capillary pressure, which reduces circulation, and therefore you will set off localized areas of reduced blood flow and therefore reduced oxygen and increased sensitivity in this area. This was further confirmed by a study by Sidkar, which is from 2009, where he used ultrasound technology to look at trigger points and the surrounding soft tissue. Here is a picture of the equipment he used, and he used an ultrasound probe with a vibration source stimulating the local area. And these are the amazing pictures that he showed. This is a grayscale image of the trigger points in the trapezius, and you can see that there is an area, obvious area, hypoechoic, where the trigger points occur. And if you look at these are smaller hypoechoic areas in the muscle which correspond to trigger points. Now, with ultrasound, you can look at blood flow, and this is using the blood flow capability of ultrasound. And what he shows is that the blood flow in the trigger point is significantly reduced as compared with the tissue around it. This correlates really closely with the previous evidence that there is increased pressure causing reduced blood flow, and this ties up the evidence of reduced blood flow secondary to increased pressure and in the local area of trigger points. Now we come to what I consider to be the most exciting recent study done about the pathophysiology of trigger points. Why are trigger points painful? Because this is really one of the fundamental questions for which people have not had a really good answer. This was a study done by Shah and Elizabeth Gilliams, and it was done in 2008. And they were looking at the biochemical milieu of myofascial trigger points using microdialysis. And basically what they did was to use this very, very fine needle, place the needle carefully down through the skin into a trigger point, and as they moved the needle, so first as they pierced the skin, they took samples of the tissue fluid right at the tip of the needle. And as they move it down, so they went through the skin, through the subcutaneous tissue, they took a tiny sample. They went down to the muscle. As they went into the muscle, they took samples. And what they were seeking for was a localized twitch response, which is shown now to be the most significant sign that you're actually in a trigger point. So they moved the needle until they had a localized twitch response, and when the needle was in that area, which is a small area, in other words, right in the center of the trigger point, they took samples again. What they did was they put the needle into two muscles. The capillary samples, these little dialysate samples, were analyzed with very, very high-tech stuff, and they used two kinds of procedures. The first was called immunoaffinity capillary electrophoresis, which is shortened to ICE, and the second was capillary electrochromatography, CEC. And what they were looking for was a number of substances that are produced in what's called the inflammatory soup. Now, this is very fascinating. When there is trauma or inflammation in your body, certain substances are produced in great amounts just locally in that area. These substances include hydrogen ion, which is acid, and we know that lactic acid and other acidic substances are produced in areas of damage, plus norepinephrine, bradykinin, substance P, and then there's some other fancier ones called calcitonin gene-related protein, tumor necrosis factor, and a whole set of interleukins. Now, what's important about these substances is that it is known that all of these substances cause pain in their own right. If you inject them into people, they cause tremendous local pain. So what they were looking for was answering the question, are more of these substances produced in a trigger point than in other surrounding areas? In their samples, in their people, they did two things. The first was they took people who had active triggers and they compared them with people who did not have active triggers. The muscle that they looked at was the trapezius muscle, a really good choice because the trapezius muscle is probably the commonest muscle to have trigger points in the body. So they're comparing, firstly, people with triggers and people without triggers in the trapezius muscle. But then they did something else, which is very clever. In the people, in everybody, they took a sample from the trapezius, but they also took a sample from the gastrocnemius, which is down in your calf. And what they were showing there is that in people who had an active trigger in their trapezius, and if their levels were high, if you took in the same person a sample from a muscle which did not have trigger points in it, would that be different? So they were looking at the levels in between people, triggers and not, but when the person had triggers, say their level was high, they were looking to answer the question, is the level high because it's in a trigger point? Or is the level high because that person happens to have high levels? And these were the results that they got, which are actually astonishing. If we look, this is the trapezius muscle, and this is the gastrocnemius muscle. In these two graphs, what they were looking at was the level of substance P and the amount of norepinephrine or noradrenaline produced. And so both of these people had trigger points. And what we see is as the needle moves through skin, comes along, and it finally they strike the trigger point, the level within the trigger point of substance P comes up. Now what they did was very interesting. They left the needle in the trigger point itself. And what they were actually doing by doing that was they were doing dry needling. So, and as we know, dry needling is actually a treatment for trigger points. So leaving the needle in the trigger point and then taking samples over a period of time, and this was over about 30 to 45 minutes, they found that the level of substance P increased and then it decreased over time and returned back to a low level. At the same time, the level in the gastrocnemius did not change because it was not in a trigger point, it was just in normal muscle. Now, substance P is this fascinating substance which was described a number of years ago, where they isolated it and it created this powder, and they gave it the name of substance P for substance powder because they didn't know what it did. And then over time they recognized that this is the most powerful algogenic substance that we create in our body. So it's substance P for substance pain. So what you see in this study is that substance P is produced in huge amounts in the trigger point itself, and then once you start treating it, it starts reducing and gets washed away. So that's the first and incredible finding in the study. The second finding is also absolutely fascinating, and that is that noradrenaline is produced in huge amounts. And noradrenaline is actually a neurotransmitter, and it is the neurotransmitter that is produced by your autonomic nervous system. I'll talk about that a little bit more, but you see here how noradrenaline increases, this little biphasic, hugely in the trigger point. It then gets washed out after leaving the needle over a certain period of time, and the gastrocnemius muscle doesn't change at all. The autonomic nervous system is the system that will have a profound effect on local blood flow to an area. So, as you know, if you feel tense and nervous, you'll get cold, sweaty hands. This amount of noradrenaline in the localized area will cause a reduction in blood flow to the area just around the trigger point and add to the problem of getting oxygen into the area. Also, noradrenaline will stimulate the nerve endings around the area and sensitize them, and thus cause increased messages going from the localized trigger point area up to the brain, increasing so-called peripheral sensitization from the trigger point itself. These are further graphs, and what we're looking at here at the top is the pH, which is the amount of hydrogen ions. So in other words, we're looking at the acidity of the area. And again, we see that in the trapezius, the acidity is normal and then it drops right down. So in the trigger point itself, there is more acid produced because, again, there is less reduced blood flow into the area, and there is a certain amount of local damage. Aerobic capacity is exceeded, and this area now is using anaerobic respiration, and therefore you make huge amounts of lactic acid. That's what this means. And it's just local, and after a while with the needle in that area, the whole effect becomes reversed. It is also known that if you were to inject tiny little bits of acid of this kind of level into muscle, it in itself causes localized pain. Again, the gastrocnemius doesn't change at all. The gastrocnemius is the control within the person, the same person. And the one at the bottom is bradykinin. This is another significant pain-causing substance, so-called algogenic substance. And you can see the levels within the trigger point, and then as the needle's left there, the whole effect changes. And the gastrocnemius, unchanged. Look, finally, we've talked about the local area where the muscle goes into spasm, where there's reduced blood flow, and we know that the increased messages go to the Cinderella fibers, which have increased pressure in them. With the increased pressure, there's reduced blood flow, there's reduced oxygen supply, and there's increased production of these local pain-causing substances, plus noradrenaline. And some of the pain-causing substances, the algogenic substances, like bradykinin, especially substance P, calcitonin gene-related factors, and noradrenaline, stimulate the nerve endings and cause huge amounts of messages to come pouring up from the local area. They also are pro-inflammatory, and they cause an ascending inflammatory process to move up the nerve cell and arrive in the spinal cord, in what's called the dorsal horn of the spinal cord. Now, in this area, so this is the message coming across. There we've got substance P, calcitonin gene-related factor, and these messages are going to be passed on to the next set of neurons which are going to go up to your brain. There is a set of inhibitory interneurons, and these normally damp down the inflammation in here. But with the localized message overload, this inhibitory system gets switched off. And what happens is that, and this is what's being shown here, that you lose the inhibition, and so you get a huge amount of messages shooting across into the second nerve going up to your brain, coming here, going whoosh. So the initial messages coming from the muscle are amplified in this area. Part of the reason they get amplified is because of the increased messages. Part of the reason is because of the pro-inflammatory messages that also come up the nerves. And this means that the original pain messages are spread up and down inside the actual cord itself. And so you now start feeling pain in other areas. You may feel it spreading down in the typical zone that you would get with trigger points, and you get the typical pain zone from trigger points. This process is called central sensitization, And it is now being recognized that this is probably as important, if not more important, in why trigger points come back and in why trigger points become chronic and last and last. So, look, what I've talked about here is the most recent work and some of the more significant previous work that make up the evidence base for trigger points. The most exciting study was the one done by Shah, looking at the localized changes that occurred just inside a trigger point as different from the other areas. I leave you with this. Thank you.